Good morning, everybody. Welcome to our coffee breaks with TRANSFAC. So grab your coffee and relax. Prepare your questions. Today we will have a very interesting topic. We'll talk about ATAC-seq and how to use TRANSFAC to analyze ATAC-seq data. Okay, let me share my screen. And we'll start. All right, so ATAC-seq. ATAC-seq is actually a very interesting method to discover chromatin structure and basically to find open regions of chromatin in different situations in very many different cells. And today I will talk about how to use TRANSFAC to analyze this ATAC-seq data, and I would recommend you to read this review about ATAC-seq, hitchhiking guide of ATAC-seq data analysis. Well, although it's maybe already 2 years, 3 years old, but it's still very good. And I will show a little bit of slides from that review, just to introduce to you what is ATAC-seq and which methods are in general are used to analyze this data. So basically, ATAC-seq is a way of analyzing epigenomic information from the cell. So there is a specific enzyme called transposase TN5, which is used to cut, areas of DNA which is free from nucleosomes. So basically, these enzymes can only cut the regions, access the regions of DNA which are empty from nucleosomes, basically open. And as you know, if this region is open from nucleosome, it can be involved in regulation of genes. And when it accesses this area, it starts cutting the DNA in pieces. And then those pieces can be sequenced with a modern sequencing technology. And then those fragments of sequences actually are mapped to the reference genome or to the genome of your choice. And then you can see basically where those enzymes were cutting the DNA and basically showing you the areas of open chromatin. And these parts which are detected by this mapping of the fragments is called peaks. And an interesting thing here, which we can also touch in our talk today, and also example which we will analyze, is that actually, of course, these open areas, especially near the start of transcription, in the region of promoter of the gene, they might be occupied also by transcription factors. And that's the whole story. So the transcription factors are coming to the open chromatin, and then they start binding to this region. And then after binding and anchoring of those regions and interacting to each other, they start regulating the genes. And actually, in this signal of ATAC-seq, sometimes you can see even those kind of valleys, which may indicate also the regions of chromatin which are closed by seeding of transcription factors. So this is also used by some of the tools to find which transcription factors are actually binding there. Although in the example I will show, we are not using this kind of signal explicitly, but we'll see it at the end. And if you look at the whole pipeline, how this data is analyzed, first, of course, you get the sequence, and then you have to map those sequences on genome. And there are several kind of steps you can do, like you have to take into account the fragment lengths in case of you have you sequence from both strands, and then there is some post-alignment processing and quality control, and then after that you see actually how those peaks are. How those reads are mapped to the genome. And then there are a number of peak calling algorithms which would take those data and we'll identify where actually the peaks are. And then there is also certain specific tools which are specifically important for ATAC-seq data. And then after you get these peaks, there is a step which is called advanced analysis, when you can actually do the peak differential analysis. Basically, if you have two types of conditions, and we'll have here today, you have actually cells in one condition and in another condition. And if you have ATAC-seq data in both conditions, then those peaks can be differentiated. So either very simple by just overlapping them, or more precise if you start counting reads in one condition versus another. It can be done on peak-based, or it can be done also other ways, and we'll see it in a moment. And then you can do, de novo motif discovery, which we are not going to do today. So that will be maybe for the next coffee break. Today we'll actually focus mainly on those last parts, also including integration with RNA-seq. When we, in fact, we will focus today mainly on motif enrichment analysis. So once you have the peaks and you have RNA-seq data, so then you would like to know what is the mechanism of all these events. So that's the whole focus of today of our talk and our seminar. And if we consider what we are going to do and what is actually approach, general approach which we follow in our tools and in our studies, and what we offer to you to try is actually so-called upstream analysis. So what it means that when we do the measurements of, for instance, of expression of genes, like we will see also today later, then and also we do kind of analysis of ATAC-seq data, so the open chromatin where transcription factors can bind. Then the question is how these events are controlled, or what is what happens upstream. Upstream means okay, so we have these genes which are differentially expressed, and obviously, or most probably, this happens because there is a certain areas of chromatin, certain areas in promoters or in enhancers of those genes which gets open, and those areas are now occupied by transcription factors, and we can detect signals. We detect motifs for these factors which are served to bind for these factors. And then, but then the next question appears. We'll very quickly go today in those areas, but basically the final answer which we want to know is what is the mechanism. And mechanism means if these are the factors which are binding to these regions of genes, regulatory regions of genes, and then they're controlling the expression of the genes, then what is controlling those activity of those factors? And then we have to go upstream from those factors and see in the signal transduction cascade what can be leading to activity of these factors. And then we can, if we find the so-called master regulator, then we basically can probably go to search for drugs which could treat those master regulators. And so from point of view, what we have in our hands and we can offer you is actually a database called TRANSFAC, which is a collection of known motifs or many different transcription factors. And so if we talk about human transcription factors, so then approximately what we have estimated recently that more than 95% of all human transcription factor have at least one motif in our database. So it's pretty good coverage of all transcription factor motifs for human genome. So that basically help to find really those transcription factors which are involved in your case. Of course, for this step of upstream analysis, you need another database, and we have it. It's called TransPass. It's a database about signal transduction network, and we talked about that a lot last time. Here, you just you should know that this is used to find the master regulators. And about the drug, there is another database which is called Human PSD. And from the data point of view, so to analyze gene expression data, we use transcriptomics, and this you see this typical RNA-seq picture when you sequence RNA and you map those reads from RNA on genome, and exactly in case of predicting binding sites for transcription factors, here we need epigenomics. And here on the picture, you see more like a ChIP-seq data when you know which transcription factor or which, for instance, modified version of nucleosome is binding to which various places. But pretty similar, you have data from ATAC-seq when you don't know which factor actually is involved. You have to guess, and that's how you will use here TRANSFAC, but you have knowledge about which regions are open and which regions are closed. And okay, on the level of pathway analysis, we probably, if we can have proteomics data to help us to find these master regulators. All right, if you go for to look around for various algorithms which help to analyze ATAC-seq data, and this is a picture from the same review paper I mentioned in the beginning. So as you remember what we said, that first you have to find the peaks. So when you pre-process the data, so you have to identify the peaks, and then once you find the peaks, you have to analyze. You have to select which peaks are important, which are not. And you see a lot of algorithms here, and we'll use today MAX2 to detect the peaks, and it's count based. Basically, it's how many counts, how many reads are found in a particular position of the genome, and then it gives us information about the peak. And actually, the same algorithm MAX2 is also detects how important in this peak, again based on the counts, how many reads are actually found on that peak, and it has a certain model, a statistical model behind, and can estimate the probability of those peaks to be by random chance. But there are other algorithms which you can read in this paper, but we will not consider today, that you can basically do the sliding window, not necessarily based on the peak, but just go through the whole area and simply count your reads. Don't detect the peaks, just go with sliding window. Or you can detect peaks using so-called shape-based. So actually, if you have those hills and valleys of number of reads, then this gives you a particular shape, as for instance, the shape, as I said, when transcription factor is occupying some place, there is a shape of a little valley. So this kind of information can be used to analyze those peaks and to predict them. And there are a number of tools which can do that. So you can find those tools in various packages. All right. So, but today we'll go to one example to actually analyze this data and to show how tools can help to analyze that data. We took it from a pretty new study. It's like November 2023 was published in Gene Expression Omnibus. The paper probably was not yet published, so at least there is no knowledge about this paper in this entry of Gene Expression Omnibus. Very fresh data, and that's about acute leukemia, acute myeloid leukemia, AML. And what the authors of this work did, they actually had a cell line of this leukemia cell line, and then they did quite a number of various high throughput experiments. So they actually, what we do now, we'll go to that entry just to show it online. So this is the entry, and as you can see, they did in fact the whole series of experiments where they did ATAC-seq. They also did high-C, and they did also RNA-seq, which is not shown here, but this is the data from RNA-seq. If you look at the whole set of data, so these are the data from RNA-seq, this is the data from ATAC-seq, and from other experiments, also from high-C. But today we'll use just RNA-seq and ATAC-seq data from this paper. And just returning back to ATAC-seq information. So in the general setup of the experiment, so what they did, they in fact were focusing on one particular protein, which is called BPTF, which in fact is a transcription factor, and we'll see it today. And they knocked out it because this this factor is known to be involved in various chromatin remodeling complexes, and when it's knocked out, then many activation complexes can be created, and many oncogenes which are actually expressed in AML are going down. So actually, this is a very good target. In fact, although it's a transcription factor which is difficult to target, but and we'll address this point today. But in fact, if you inhibit this factor, the AML cells, they start getting into apoptosis. So they actually, you can cure a sort of acute myeloid leukemia by doing that. So, and we'll use this data to compare. So, and actually a very good, we can of course take the data, already pre-processed data from this entry, but in this paper, they actually use this sliding window approach, although I would like to show how to use the peak calling approach. That's why I went directly to the raw data, and that's you do by clicking on this SRA run selector in the National Library of Medicine. There is actually which consists of raw data, and in a special SRA format, where you can see these are the samples from wild type, these 4 and 5, and also 10 and 11. So you will remember that these are wild type, and these from 6 to 9 are the actually the entries for the knockout experiment. So and now we will go to our tool, and actually because I know these are huge files, like these are like 1 GB and these like 2 GBs in the compressed form, but if you are decompressed, it's like up to 8 GB size of the FASTQ file. So it takes a lot of time to upload and everything. So I will show you the already pre-processed uploaded steps, but just to mention that what we do actually, we go to our tool, which is called geneXplain platform, and you've seen probably this tool already in my previous coffee breaks. This is really a platform containing a lot of algorithms and tools for various types of omics data. So starting from RNA-seq and chip-seq and generally epigenomic, and we will talk in details about this topic today. And what we first have to do is to upload my data, and this button opens for me this this tab where I can see my data. So you see a lot of data here. Let's first close it. So this is a project which I created here, and in the data folder I have several files. Basically here in the FASTQ folder, I showed how I uploaded these 2 files, the FASTQ files. The uploading is described in our manual. It's when you start from SRA, basically what you do, you copy SRA accession number, and you go here, you click here, and then you actually have to put it in a raw format. And you put here the SRA accession number, and you press OK. I will not do it now because it's quite a long process to upload this. But what it happened, it automatically will go to SRA archive, download this file in SRA format and automatically convert it into FASTQ format. So, and that's exactly what happens with these two files. Actually, this is one SRA entry, which contains two FASTQ files because this is experiment done from a double-strand sequencing from both strands actually, and these are two FASTQ files. All right, so and then we have those FASTQ files, and now there's a lot of processes of pre-processing it and actually also quality control. We will not do it because it's not topic for today. I will just show you the final BOM files, which are done by our tools when we map those FASTQ files on genome. Basically, to do that, again, I show you quickly to you that what happens in the analysis tab. We have here the Galaxy tool set, and there are a number of tools to work with mapping of genome reads. So here we used actually, let me see which one we used. Right, so I think it was mapping. Right, so I used the bow tie tool. So and again, so this is a tools which we imported from Galaxy package. So our platform is naturally linked to the Galaxy format. So any tools from Galaxy, and we basically put a lot of Galaxy tools here. So, and this bow tie, you can specify here whether it's single end or paired end. And if it's a paired end, then what you do, you take your FASTQ files and put it here by drag and drop. And here you have to specify the reference genome, which is in fact the latest one, the ensemble 38, so the latest genome. And there is a number of parameters you can define. We can stay with the default parameters, or you can actually specify more parameters here to do the mapping in the best way. So for that, you have to have a certain knowledge or read about that in literature, but here you have those parameters available, and then you have to give your output file. So this is a 6, right? So we put it here. Well, we have done it already, so again, I will not run this, not to overrun this file. So, and then you simply click run, and then you wait. And then the bow tie algorithm, what it does, it takes all the reads from FASTQ file and start mapping it to the genome. All right, so once they are mapped, and this is actually the BOM file which is created, you can see what is the size of that file. And actually by here in info box, you see which files, the size of the file, and how many line reads here, and how many non-aligned reads, yeah, quite number, and you have the size of the file. So now what you can do is also you can in fact look at this file on the Genome browser. So you drag and drop it here. So I have do I have it? No, I don't have it here. So you drag and drop it here. And then you see this file now here. And in fact, in that area which we are looking now, and I will talk about this area a bit later, there is very few peaks, very few reads. These are actually they demonstrate the reads in that area. Although in the other example, this one is after knockout of the transcription factor. And the wild type actually contained in that area quite huge peaks. So that's the one of the interesting topic today. All right. So you can see this BOM file. And then the next step, as you remember, is the peak calling. And as I said, the peak calling was done with a tool called MAX2. And here we have these peak calling algorithms. Well, in the Galaxy tools, we have a sizer. It's another very nice algorithm, but in this case, we used the MAX2. And the MAX2 is our tool which we integrated inside the platform, even on the level of very basic level. And then you can find it here in NGS. So we have this MAX of different variants, the newest one and the previous one. So, and again, what you have to put here is the track. So, for instance, this BAM file. So we put all the reads which were mapped on genome, and then you can put a control pick here, control track if you like, and which I didn't do here. And then there again, so you can specify parameters. For instance, there are parameters which defines how big is enrichment, or you have to expect, and what are the parameters of the statistical model behind, and then you do it. Yeah, so you run it again. So which I did, and here the peaks, and again you can see that it found 188,000 peaks in the genome for this whole one experiment. So again, we can look at this peak where they are. So if you simply drag and drop here, so for instance here you don't see the peak because there are very few reads. But if you take this one from 11, for instance, and put it here, you see that peaks. This one particular peak was detected because here you see a lot of reads. All right. So the peak calling algorithm works, obviously, and you have finally the peaks. All right. So, okay. So while I'm talking, please prepare your questions. Any question to me? If some some steps are not clear, or you need more detailed explanation, or maybe you have a question about some other tools, just put it in Q&A section, and I can try to answer it maybe at the end, or if it's a question concerning the current step, I can answer it right away. All right, so we have now peaks, and what is next? So next is to analyze those peaks and to find enrichment of transcription factor binding sites. And what we are talking about. So just briefly, we can return to our PowerPoint, and just to remind you, so there are two things. One is we search for binding sites for transcription factors using so-called motifs from TRANSFAC, which are presented in the form of positional weight matrices. And those positional weight matrices were collected from different literature, or we constructed them by ourselves. Our team constructed it by aligning known sites for transcription factor and then building this weighted positional weight matrix. And then using certain formulas of scoring, we can apply this matrix to search for new sites. And then if the score of that hit is higher than a particular threshold, then we claim that's a known site. So, but this way we can find just individual sites. To find the enrichment of the sites, we have to count how many sites we found in the promoter. So here we show just one, but if you found many sites, and if you have not just one promoter, but a number of promoters, what we do, we count all number of sites found on all promoters, or well, if you talk about promoters, but in this case, we are talking about areas of ATAC-seq peaks. And we talk about this, we analyze these ATAC-seq peaks and calculate how many sites of each particular, for each particular positional weight matrix we found in those areas. And then we compare it to similar area in genome, but which are not detected in the ATAC-seq experiment. And then using statistics, we can predict whether this motifs is enriched or not. So we go now to see how that happens. So here we have what we did. Actually, what I did, we have 4 experiments on wild type cells and 4 experiments on after knockout. So to have a really collection of all the peaks together. So I actually joined all the peaks. So in here, there's a tool for joining the peaks, join peaks. So this tool is called join tracks in our analysis tools. And you simply, what I did, I put here all the peaks detected in each experiment from the wild type, and I put them together. So if you put it here, you see these are the peaks coming from all these experiments. Then actually what I did here, I pre-processed those peaks and cut actually all this collection of peaks are now presented by one joint peak, which is covers when the peaks are overlapping, and if peaks are just found by one of the experiment, it also adds to that. So it collects all areas which are covered by all experiments in wild type cells. So at the end, what I get here 74,000 peaks in the whole genome. All right. So, and now what I want to do, I want to analyze those peaks from a point of view of the motif enrichment. But first, what I did, I actually intersect those peaks with a similar set of peaks which are coming from knockout experiment. Because what I want to know, I want to see those peaks which are found in the wild type, but not found in ATAC-seq experiment. So because there are some peaks which are found in both experiments, I mean in the wild type and knockout, right? So I want those which are found in wild type, but not in knockout. And other way around, I want to find those which are in knockout, but not in wild type. So for that, to do that, I have this another tool which is called intersect tracks. And it found those peaks which have just difference, so which are only present in the wild type but not in knockout, and there are only actually there are only 22,000 of such peaks. Interesting, right? So out of 400,000, only 22,000 are different. So it means majority of peaks, in fact, are the same before knockout and after knockout. But only 22,000 are really specific, and I want to analyze those specific ones. And to do that, I have actually my tools to analyze epigenomics data. And here we have a workflow, discover, function, analyze genomic intervals, and site search with TRANSFAC. And here we have this most advanced pipeline, version 3, which is working with TRANSFAC database. And those who haven't seen yet our interface, just you see what happens here. It's not just one tool, it's a pipeline. We call it a pipeline of workflow, which combines several different tools in the pipeline. So, and here you see below this pipeline. So as you can see, what happens, you input the track, the peaks, and then immediately those peaks are started TRANSFAC match. So basically it takes the whole library of matrices from TRANSFAC and analyzing sites in these peaks. And also it creates random tracks. So as a background, we have a process of creating a number of random tracks, which means we simply randomly pick up regions from genome and use it as a background. Because what we want to see really enrichment. We want to see those sites which are happening here, and very many of them, and not, or very few of them in the random tracks. So that's done in this pipeline. So then those results, so sites found in our track and sites found in the random track are compared, and then we'll see the result. So, and how to do that? Basically, you simply drag and drop it. Again, here you have to define the species, and here the sequence source is already defined. Annotation source from ensemble database And here what you need is to put the so-called profile. And the profile is a set of positional weight matrices from TRANSFAC, which we want to analyze. And that's, well, by default here, we have this non-redundant small set of matrices, we just for the quick search and to see the major signal. But actually, if you really want to do a big analysis to find all potential enriched motifs, you would select here, this, for instance, is a vertebrate human P001, which means these are all matrices from vertebrates species. So it's around 7,000 motifs, and the optimization of the cutoffs of those motifs is done in such a way that they guarantee at least one hit by 1,000 nucleotides in random place in genome of human genome. So that's a good level of cutoff from which we can start to search for enrichment. All right, so then you specify this, and then you click run. I will not do it right now because it takes again quite a lot. But what we'll do now, we'll see the results. So the results are put automatically in this folder, which you can of course change the name of that folder. So this is a TRANSFAC analysis, and here is a number of files. First important file is the site search summary. So that's a summary of motifs which are found enriched in the peaks, in these 22,000 peaks of which are, again, these are peaks of ATAC-seq in a wild type cell of AML. So actually this is the cancer peaks, which are very specific for cancer because we don't have those peaks which are happen after knockout of that very important factor, which actually kills the cancer. All right. So this 22,000 cancer peaks. Okay. Well, it's maybe not the right word to call leukemia cancer. It's more like a, although, yeah, that's more terminology thing. Anyway, so here you see the list of 969 motifs which were found significantly enriched. So quite a lot of motifs, actually, I must say. And you can sort them by the p-value of enrichment, and you see it's pretty big. Or you can sort it by actually yes to no ratio. It's in fact the enrichment score. It tells you how many times the frequency of those motifs in the peaks are higher than in the random peaks. And for instance, at the top we have this STAT5, which is happening 14 times more frequent, although by itself is not very frequent. It's like only two sites per 100 100 peaks, sort of. So there are few sites in those areas of STAT. But actually STAT5 is a good hit. If you search for AML and STAT5, you definitely will find that STAT5 is known very important for AML pathology. Actually, what we are interested here to look whether the motif for that factor which was knocked out can be found here. Why? Because what happens, these are the peaks, and we know this factor is involved in building this chromatin, complex, activation complex in that area. And when we knocked out this factor, it's not binding any longer to those regions, so they're depleted. That's why these areas maybe contains those motifs for that factor. So which factor we are talking about? Actually, this is a factor which is called, there are various names for it. It's called FAC1. So that's the same name which of that factor which was knocked out in that experiment. And we have one motif for this factor in TRANSFAC. I have actually this motif somewhere on my screen. I think I had it open. Let me see. Yes, it's here. bring it up. All right, so that's motif. It's very A-rich, A and C sometimes reach motif for this factor, FAC1, and in fact, you remember that was BPTF, so that's another synonym for that factor. This particular motif is in TRANSFAC and it was created from actually artificial experiment from Selleck's experiment, but still this motif is pretty good to search for binding sites for this factor. And all right, so we simply have to search here in this table for factors. The motif is a name FAC1, and we search for this in ID. So we could one. All right, so we have actually this factor, and you see the p-value of enrichment is pretty significant. It's p minus 22, and the yes to no ratio is not very high. It's 1.2, but actually these sites by themselves is very frequent. But still, you see it's more frequent in my peaks compared to random. So the p-value of that enrichment is significant. We can see those areas actually when we click here on this button. We can it generates this table, this one, which shows each peak here as one line, one bar actually. This is ID of that peak, and well, actually, I annotated them also by the positions in genome. So here, this is a chromosome 6, and from this to that, so the length of that peak is 639, and this peak, as you can see, is very interesting. It's actually have this number of motifs in the area for this factor. And they kind of very many of them in that area. So in fact, we can have a look at that area more details. That's why I opened that here, and you can see here actually what we see is that peak, and these are the motifs for this factor. A lot of A, in fact, and you see here A. It's more or less a poly A track, and it seems to attract a lot of factors in that area. And that triggers those ATAC-seq experiment, because this area was opened because of these factors, and these factors occupied this place. Interesting, we actually see also this valley here, because you see here, if you zoom a little bit down, for instance, in this knockout, in the wild type experiment, you see the peak of the reads. Real peak of the reads is a little bit left of that area of the factor binding, and a little bit right. So you see these factors by binding in that area, they in fact occupy the place, and it doesn't allow this restriction enzyme to cut. All right, so by doing this, you see we can find a lot of places where those peaks of those motifs are enriched, and we also see which motif for which transcription factors are enriched. So we also have here in the output the list of transcription factors, namely 292 factors which whose motifs are found to be enriched in that areas of peaks. All right, so that's very detailed. We show very detailed how you can analyze motifs, simple motifs in your peaks. But I wanted to show you a little bit of story here, also showing the whole mechanism. And I cannot go too much in details in that. I will just show you the final result. As you remember on my slides, once you found the motifs, it's only the half of the story. So now you have to think how those transcription factor might be activated through maybe signal transduction pathways, and maybe you can find even the master regulators in these pathways. And that's exactly what I did. And I did it actually using another tool, and we have here the tool called Genome Enhancer, which changes completely the whole interface. Although this tool is a kind of a layer
above the geneXplain platform. It gives a very easy interface to start the analysis. And here what I did, I took actually both these ATAC-seq data, and I also took gene expression data from the same cell. So if you look here, so you can have a look at the results. So it's now we'll show a bit in the screen. All right. Yeah, let me open the results first. So this tool allows you to put various omics data in the experiment, in the tool, and then the tool will combine this omics data together and analyze it. So what we did here, as I said again, so we took actually these two tracks, we call them tracks. So this is a joint peaks from wild type experiment, and these are the joint peaks from the knockout experiment, and drop them here in gray box, basically like that. So of course they are here already. Then we have here the RNA-seq data, which is in fact a very simple data. It's a table where we have all 56 ensemble genes, 56,000 ensemble genes, and then we have already optimized or actually normalized counts for each experiment. So these are the counts of RNA. So basically they show the RNA-seq. So what I did then, I took this table and drag and drop it here like this. And then you see each of the sample here. So then we press next. And then in this field, so it takes a bit time to get this information. We can formulate which experiment and which control we are going to use. So it takes a bit time. Yes, here we are. So we have this AML condition, and we put those RNA-seq experiments here, and also the ATAC-seq peaks here. And then we have AML after knockout, and we have here RNA-seq experiment and the peaks here, right, from the knockout. Then we press next, and then that's the only entry to start. So what we do here, we first define the disease. This is a leukemia, so which we select, and actually more specifically leukemia myeloid acute. So we have these two types of disease, and then we select here two conditions to compare. And we want to see, actually in this experiment, now what I did, I compared the wild type compared to the knockout. So to want to see what genes are actually expressed in wild type, so oncogenes, and which ones then goes down when you do the knockout. And then I simply start the analysis. It took maybe 2-3 hours, because actually doing a lot of also the things which I didn't talk about, is like it's also using genetic algorithm to find the combinations of factors. But at the end, what it gives you results in the form of a report. Here I have several of those runs. Let me see which one I want to see. I think that was which one, I guess. All right. So that's this is a report which finds the major master regulators, and also reports which transcription factors were the most enriched in our tool. So I will, you can have this report when you run it. Basically, it also compares the expression values of RNA-seq, but for us, what is interesting is the result of the motif search. And that's present here. So what it does, it now takes the peaks of that wild type and intersect them with upregulated genes. It now analyzing not the whole set of peaks, but only those peaks which are present in vicinity of upregulated genes. So we, with this, we kind of propose that these are enhancers. These are areas which may be involved in enhancement of expression of those genes. That's the first thing. And the second is actually looking not just for the single motifs, but it looks for a combination of motifs. And that's what you see here. It's a combination of motifs which it found. And that's how the areas, those peaks, which contains this combination of motifs, as discriminated from other peaks which are actually in random places of genome. So they although quite overlapping, but this difference is significant, and AOC is okay. So it tells you that this signal is pretty noisy, but still you see that this combination of motifs is really present there. And of course, you can see this combination by clicking here. So we can sort it by the CMA score. It's called the CMA score, which tells you actually CMA score is this one. So this is a score. So the higher the score, the more enriched your peaks by this combination of motifs, right? So for instance, this peak, which is in the upstream 5' region of cast gene, has a maximum CMA score, and this is a combination of binding sites which are found in a cluster inside that peak. We can see that also in genome browser. So if we have more time, we can have a look at that. And then these are other genes with their regulatory areas. Basically, by doing that, we define the enhancer regions of those genes. Because on one side, this is an area which is open. It's an area of open chromatin. And on another side, it contains the cluster of motifs which work in combination together to regulate that gene. So that's what you see in the report. And actually, if the same you can see for the downregulated genes. In the downregulated genes, you see a different combination of motifs. And one thing which I want to show to put your attention on is if you have a look, you see here many important transcription factors. But for instance, this one, it's called YY1, is actually present here twice in both modules. So these are modules, this kind of part, two modules of one composite model of that enhancer. So it seems a quite important motif. And if we see that in the genome browser, so we can go here back, and if we open that in platform, we can have a look in those sites which are found in that experiment. So they are quite deep in the tree, but I know where they are. So actually, this is the result of that combination analysis, and these are the sites which are found here. Actually, this one track which we are looking here is not really here. It was not near any gene which is upregulated, right? So although it's a very important track, an important peak, where a lot of these factors are binding and its chromatin is open, but this area is not probably involved in regulation of genes. It's just a trap for many factors. So, but if you look at the vicinity of genes which are upregulated, you find maybe smaller peaks like these ones. So these are in the vicinity of which gene actually? Let me have a look. This is a do we have here genes? Yeah, this is a gene which obviously was upregulated, and in the upstream area of that gene, we see. Okay, I probably should remove many tracks here, but keep only those ones which I need. All right, so this is a, yeah, this you see this is a gene. This is a peak of the joint peak of all experiments, and these are the sites found in that peak in nearby. And you see what I wanted to show you. This is a definitely cluster of sites, and these YY sites are present quite in many, and it actually at the left part and in the right part, and that's very common. When I looked through this results, it was very common feature that those areas are actually located where you see YY sites, YY1 sites are at the borders of those peaks. And that actually triggers me to look at these YY sites more in depth, and actually was very, if you look at the YY factor, it's a very interesting factor. It's called Yang factor because it has a controversial regulation. It may regulate upregulation and downregulation. It may be involved in tumor progression or tumor repression. So it has many roles. And even in very early papers from actually 1996, they found out, or they proposed, that actually this factor is involved in DNA bending. So for instance, in this case, you have a promoter and TBP, and this factor NFY is involved in that activation of that promoter. And if you have this YY1 site and this factor is binding here, it brings this DNA closer to activation complex, and this triggers the genes to upregulate. But if you put the DNA a little bit different orientation, it's actually breaks the activation. So these experiments show that, in fact, that's this factor YY1 is involved in DNA binding, and the current, when we look at the modern papers from the last years, you can see that they show that these factors actually work as an oligomer. That's why you see many sites nearby. So they help this factor to close together to each other and create the complex. And they're involved in the bending and in looping of DNA and bringing actually enhancer regions to the promoter and increasing the expression of those genes. So there's another paper here, for instance, to show that this Y1 factor are playing the similar role like CTCF. CTCF may be on a higher level of chromatin, but Y1 and really an enhancer promoter loops. So that's, and we actually see those features here. And if you look, when I looked at YY1 and actually AML, leukemia, very clear and very early, it was found that that's important factor controlling leukemia. Again, if you knock out YY1, these leukemia cell, they also go into apoptosis. Likewise, the factor which was analyzed in that paper. And the last thing I wanted to show today is a very quick glimpse on the final master regulator results. So after we found these factors, the tool goes further and it builds such a network which connects the upregulated genes, which we found here in RNA-seq, with those transcription factors, which are shown here blue, whose motifs are found enriched in the ATAC area near those genes, and then it builds the master regulator network upstream, and then defines the master regulators, and among them this FIN kinase, which is again, when we look at in the literature, we found that FIN is indeed very important for the IML mechanism. So, and actually in the same report, you also see here which drugs are involved, can be used in leukemia, and prioritized here according to those master regulators like Yin and Fin, which were found here. And this drug is known to be inhibiting those master regulators. So, but that's more like a drug story. What I wanted today to show you today that this analysis of ATAC-seq data is just a first step, and then you can even go further to find master regulators. All right. So with that, I think I showed what I wanted to show today. Anyway, so thank you for attending, and we'll see each other next time. Okay, see you, and goodbye.